Many molecules studied in biology are based on carbon. The chemical properties of the carbon atom allow it to form many diverse structures with a range of functions. Carbon is the sixth element on the periodic table with 6 protons and 6 electrons, and 4 of the 6 electrons are in the outermost electron shell called valence electrons. One of the most important chemical properties of a carbon atom is its ability to form 4 covalent bonds with other atoms because of the arrangement of its 4 valence electrons. A covalent bond is formed by the sharing of valence electrons between 2 adjacent atoms. Covalent bonds are the strongest type of bond between atoms, and therefore carbon-based molecules are very stable. Carbon atoms can form covalent bonds with other carbon atoms, or with atoms of other elements such as hydrogen, oxygen, nitrogen, and phosphorus. It can form 4 single covalent bonds with one other element, as it does in methane, CH4, or include double covalent bonds as found in carbon dioxide, CO2. Carbon atoms can also link up by covalent bonds to form a chain of any length, like in fatty acids, or a ring containing carbon and nitrogen like in the nitrogenous base thymine. Carbon's ability to form 4 covalent bonds allows for a large range of simple to complex molecules to be formed, making it a great building block for all living things. Macromolecules are molecules composed of a very large number of atoms. The large, complex molecule is often referred to as a polymer composed of smaller subunits called monomers. The main classes of macromolecules referenced in IB biology are primarily carbon compounds such as polysaccharides, which are sugars, polypeptides, which are proteins, lipids, and nucleic acids. Each of these is made by linking respective subunits, monosaccharides, amino acids, and nucleotides, via condensation reactions. In a condensation reaction, 2 molecules are linked together, and in the process, a smaller molecule, water, is released. This happens by removing a hydroxyl group, OH, from one molecule and a single hydrogen atom, H, from the other molecule, creating H2O. A bond is then formed to connect the 2 molecules where the atoms to make the water were removed. Energy is required to construct polymers by condensation reactions and is supplied by adenosine triphosphate, or ATP. For the IB exam, you need to know how these condensation reactions are used to build all 4 macromolecules. We covered nucleic acids in section A1.2. We will cover polysaccharides and lipids in more detail in this video, and polypeptides will be covered in section B1.2. Just as we can build up large macromolecule structures via condensation reactions, we can also do the opposite to break them down. This would mean taking the large chain polymers and breaking them apart into their individual subunits, or monomers. This type of reaction is called hydrolysis and works in opposition to condensation. Recall that the condensation reaction used to build long-chain polymers has molecules bind via the creation, or condensing, of a water molecule. To undo this reaction, via hydrolysis, a water molecule can be split and the subsequent hydrogen and hydroxyl group can be reformed on the original monomers, thus breaking the bond that linked them together. This makes sense when breaking down the name hydrolysis. The suffix lysis means to break down and the prefix hydro means water. So we can translate the literal meaning of the word as to break apart with water. And this is exactly what we see, a polymer being broken down back into separate monomers via a water molecule. Now in this video, we will cover carbohydrates and lipids, and we will begin with carbohydrates. When you hear this term carbohydrate, the first thing that should pop into your head is sugar, because carbohydrates are nothing more than sugar molecules either seen individually or linked together in a chain. The basic subunit of a carbohydrate is a monosaccharide. Mono prefix meaning one, and saccharide suffix relating to sugar. These structures are built using only 3 different atoms, which are carbon, hydrogen, and oxygen, and have them in specific ratios. Glucose, for example, is a monosaccharide, or a single sugar. It contains 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms. So we notate the chemical formula for glucose as C6H12O. H12O6. Now, the ratio rule for carbohydrates is that for every carbon you have, there are the same amount of oxygen atoms and twice as many hydrogen atoms. So we can say the ratio is 1 to 2 to 1 with carbon, hydrogen, and oxygen. Monosaccharides can come in two shapes, which we call ring structures and straight chain form. So if we look at glucose here, we can see this is the ring structure and this is the straight chain form. Both versions carry the same chemical formula. Along with the structural representation, you also need to be able to identify different types of monosaccharides that we call pentoses and hexoses. Pentoses contain 5 carbon atoms, as seen here with ribose, and hexoses have a 6-carbon structure like we see in glucose. In any case, both of the molecules keep the ratio of carbon to hydrogen to oxygen, like glucose with C6H12O6, and ribose having the chemical formula C5H10O5. You can see that the ring structure is different, with ribose having 5 points to the central ring where glucose has 6. But the ring structure is not a defining characteristic of labeling it as pentose or hexose. It solely depends on the number of carbons. In addition to structures, you need to know a few properties of monosaccharides, and specifically glucose because it is a very important sugar structure for living things. First, monosaccharides, including glucose, are small and soluble in water, which means they can easily be transported in living tissue because living tissue is based around water. Second, the oxidation of this molecule, which describes how electrons can be transferred from the molecule within a chemical reaction, can yield a great amount of energy for cells, which we see in the process of cellular respiration, but more on that in another video. So for now, just know that monosaccharides, like glucose, can be used as a quick energy source for living things. Lastly, glucose and many carbohydrates are very stable molecules, which make them great candidates for energy storage, which we will talk more about on the next slide. Be sure to memorize all of these key properties of glucose for the IB exam. A monosaccharide is a single sugar molecule, and if we connect 2 of them, we call it a disaccharide, prefix di meaning 2. And if we continue to connect sugar molecules to make a longer chain, we call it a polysaccharide, the prefix poly meaning many or more than 2. There are 3 broad categories of these large carbohydrate molecules that you need to know about, which are starch, glycogen, and cellulose. Starch and cellulose are only made in plants, while glycogen is made in animals. And looking at a few structural differences, we can see that cellulose structures are unbranched, starch structures are moderately branched, and glycogen is heavily branched. And if we look at the similarities between these molecules, we can see that all of them are made out of the same repeated structure, glucose. Before we talk more about these large molecules, let's learn a bit more about the structure of glucose. We know that glucose is a single sugar molecule that has the chemical formula C6H12O6 and commonly, but not exclusively, takes on the shape of a hexagon. So if we look at a drawing of its structure, we should be able to account for all of these atoms. We start naming carbons from the carbon on the rightmost point, which we call carbon number 1. We circle around moving clockwise to count the other carbons, but notice that carbon number 6 is not part of the hexagon structure. It branches up and off of carbon 5, which leaves the 6th point of the hexagon occupied by an oxygen atom. In most drawings within organic chemistry, assume that any corner or point where 2 lines meet is a carbon if it is not otherwise labeled. Next, we can count the oxygen atoms, and we can see that there are 6 total. Finally, we should have 12 hydrogen atoms. All of them are shown here, but know that on some structures, if nothing is shown to give carbons the 4 bonds it makes, we should always assume that a hydrogen is present to get it to make 4 bonds. Okay, so back to glucose. All of the atoms within our chemical formula are accounted for. Now, the last thing we need to know about glucose, which is very important, is that it comes in 2 different ring forms, which are called alpha and beta. There is one very specific difference that exists between these forms. Take a second to see if you can spot it. If you notice a difference between the orientation of the bonds coming off of the first carbon, you got it right. In alpha glucose, the OH group is oriented down, and in beta glucose, the OH group is oriented up. And again, this only applies to the hydroxyl group off of the first carbon. You can see that carbon 2 is always down, carbon 3 is up, and carbon 4 is down. An easy way to remember this is that beta follows the up-down pattern, while alpha follows the down-down, up-down pattern for the OH groups. Let's take a look back at our larger polysaccharides to see why these structural components are important. Starting with starch, we can see that the chain of glucose molecules contains glycosidic bonds that connect carbon 1 of one molecule to carbon 4 of another molecule. We call this a 1,4 glycosidic bond. If you look closely, you can see that all of the oxygen atoms on the 1 and 4 bonds are pointed down, and all glucose molecules are oriented this way. This tells us that starch molecules only contain alpha glucose, which is very important to remember. Only alpha glucose, not beta glucose. We can break this down further into different types of starch molecules based on if the structure is linear or branched. If we only have 1,4 bonds between alpha glucose, meaning the molecule is one straight line of sugars, we call this amylose. But we can see here that sometimes the structure branches, and this branch shows a bond between carbon 1 of one molecule and carbon 6 of another. And you guessed it, we call this a 1,6 glycosidic bond. If that happens and new chains of alpha glucose branch off an existing chain, it is no longer called amylose and instead called amylopectin, which is a combination of 1,4 and 1,6 bonds. Next up, we have glycogen, which is similar in structure to amylopectin, but it tends to branch more and is created inside the body of an animal. This structure, because it branches, also contains a combination of 1,4 and 1,6 glycosidic bonds. In addition, you should be able to deduce that only alpha glucose is used to create this structure, not beta glucose. Both glycogen and amylopectin are very large molecules that can store large amounts of energy in plants and animals, respectively. Their large structures make them less soluble in water compared to single monosaccharides, which is good for being kept as storage. But when the time comes, hydrolysis can break glucose molecules off of the chain, which can be moved to cells where energy is needed, which was the point of storing it in the first place. Finally, we come back to cellulose. Now, the cellulose you can see is very different in structure because it is made up of beta glucose only. Because the OH group on the carbon 1 in beta glucose is up and the OH on carbon 4 is down, when these beta glucose molecules join together, every other molecule has to be inverted, which we can see here as we count carbons counterclockwise. Every other one is upside down. This makes the cellulose structure unique for many different reasons, one of which is being able to form microfibrils that have extremely strong tensile strength. These are the basis of plant cell walls and give plants their firm, sturdy structure to stand upright and not burst under high amounts of internal pressure, all because of the beta glucose produced structure. And as always, for the IB exam, make sure you know the differences between all of these long-chain polysaccharides. While carbohydrates can create many large structures on their own, they also have the ability to bond with other macromolecules. One example of this is a glycoprotein, which can be found on cell membranes. The term glyco is in reference to the sugar part of the molecule, and the protein part speaks for itself. Embedded within the phospholipids of the cell membrane, we can find these glycoproteins, which contain a short chain of monosaccharides that we call an oligosaccharide. Cells can read these chains on other cells in the process of cell recognition, which is important for cells in multicellular organisms to be able to identify other body tissue from foreign cells or pathogens. A specific example of this can be seen in our red blood cells and the determination of the ABO blood groups. As humans, we have 4 different possible blood types, which are A, B, AB, and O. These blood type combinations are made by different oligosaccharide chains which are called A, B, and O. The base of these chains are identical, with A having an additional N-acetylgalactosamine and B having an additional galactose. The purpose of these glycoproteins on red blood cells are to signify if foreign blood has entered the body. So if you had blood type A and received a blood transfusion from type B blood, your body would reject the blood and it could clot. Type O blood is unique because it does not cause any rejection problems as it has the same base structure of the other two, just without the determining last sugar molecule. Our body will not recognize it as foreign and therefore not reject it. We will cover the workings of ABO blood types in more detail in another video. Moving away from carbohydrate molecules, let's now spend some time focused on lipids, another large macromolecule found in the human body. Lipids are made out of carbon, hydrogen, and oxygen, but do not follow any specific ratio like carbohydrates do. A general property that we need to know about lipids before we take a look at their structure is that lipids are generally hydrophobic. This allows them to easily dissolve in nonpolar solutions, but not in polar solvents like the water that makes up most of our body. So we have different methods of transporting them. Some commonly occurring lipids that you should know are fats, oils, waxes, and steroids. Let's take a look at the structure of lipids in more detail. The main structure of most lipids are called fatty acids. Each fatty acid molecule starts with a carboxyl group, which has a carbon atom that is attached to an oxygen with a double bond, and another oxygen with a single bond that is also attached to a hydrogen, which we call a hydroxyl group. This is often written as COOH. From there, a fatty acid then has additional carbons attached to the first carbon. How many carbons should there be? That depends on the type of fatty acid. Some have many carbons and are quite long, while others are on the shorter side and only have a few. There are two important lipid structures that are created with fatty acid chains that you need to know of, which are triglycerides and phospholipids. Both of these start with a glycerol group, which has the chemical formula C3H8O3. To make a triglyceride, 3 separate fatty acid chains undergo condensation reactions with the open hydroxyl groups on the glycerol, resulting in a final triglyceride structure that looks like this. The creation of the phospholipid is the same, except one of the fatty acid chains is replaced with a phosphate group. Both of these molecules are core lipids seen in the body. Make sure to know their structures as they will pop up many times throughout the IB biology curriculum. Taking a closer look at a fatty acid chain, you will notice that each carbon within the fatty acid chain has a hydrogen molecule attached to complete all 4 bonds that carbon can make. If there is a single bond formed between each carbon in the fatty acid chain, we call that a saturated fatty acid. You can think about it being saturated with hydrogen, because single bonds means more open spots for hydrogen atoms to bond to the valence electrons that each carbon atom has. If there are any double bonds between carbon atoms, we call this an unsaturated fatty acid. Monounsaturated would have just one double bond in the chain, where polyunsaturated would have more than one. This double bond can create a bend in the molecule, which is not shown in this image, it's actually shown in this one, and also contains less hydrogen atoms because carbon can only make 4 bonds, each double bond counting as 2 for both of the attached carbon atoms. When we take a closer look at unsaturated fatty acids, we can distinguish between 2 different isomers, or types, based on where the hydrogen atoms are in relation to the carbons that share a double bond. If both hydrogens are oriented in the same direction, as they are in this image, we call this a cis fatty acid. Cis sounds like sisters that are close to each other and have a good relationship, which is one way you can remember it. Notice how this impacts the shape of the chain, causing it to bend. If the 2 hydrogen atoms are oriented in different directions, or facing opposite sides of the molecule, it is called a trans fatty acid. This does not have the same impact on the shape of the chain compared to the cis isomer, as seen here, and results in a straight-chained fatty acid. Lipids that contain straight-chain fatty acid tails tend to have higher melting points because they can be packed tightly together. An example of this would be butter, which is solid at room temperature. If lipids contain many cis double bonds and have bent fatty acids, they tend to have a lower melting point and are liquid at room temperature, like oil. This is because they can't sit neatly and packed together next to each other because of the bent fatty acid chains. When talking about lipids, which are fats, we have to mention the type of fat that humans carry in their bodies. Body fat is a necessity for survival because it holds important energy reserves that the body can use as needed. The fat tissue that we have in our bodies is a specific type of tissue called adipose tissue, which are basically cells that are stuffed with triglycerides. Triglyceride molecules are well suited for long-term energy storage because they are chemically stable, hydrophobic, and can store much more energy than a carbohydrate per gram. In addition to that, triglycerides are poor heat conductors, which means they can help animals hold in body heat to stay warm and act as an insulator to combat cold weather. They are also liquid at room temperature, which makes clusters of adipose cells shock absorbent, which is a reason we find a lot of fat tissue around our kidneys for protection. Hydrophilic molecules are polar and attracted to water because water is also polar. Hydrophobic molecules are nonpolar and therefore not attracted to water and more attracted to other nonpolar molecules. Phospholipids are unique because they contain parts that are both hydrophilic and hydrophobic. If a molecule contains both of these properties, we call it amphipathic. Because of this property, when phospholipids are placed in water, the heads of the molecules, which are hydrophilic, are attracted to the water, and the tails, which are hydrophobic, repel the water. This naturally creates a formation where the tails of the phospholipids will be next to each other, because they are then away from the water, which creates a unique double membrane structure. This leaves the hydrophilic heads of the phospholipids coming in contact with water molecules, which is preferred. This double membrane structure, called a lipid bilayer, is what cell membranes are made of. And more on that in other videos. Steroids are lipid molecules that have a unique structure which differ greatly from the usual fatty acid chain lipids we are used to seeing. The telltale sign that you are looking at a steroid molecule is that you see 4 carbon-based rings that are fused together, which consist of 3 cyclohexane rings and 1 cyclopentane ring. You can see examples here with testosterone and estradiol. Steroids have 17 total carbon atoms within their ring structures, and can have additional carbons branching off the rings as well. Because these molecules are mainly made out of carbon and hydrogen, they tend to be hydrophobic. This allows them to be able to pass through a lipid bilayer and even stay within the bilayer, which, as we discussed on the last slide, has the hydrophobic core region where the fatty acid tails of the phospholipids come together. Make sure to remember this structural form of steroids because you will have to identify this general structure on the IB exam.